Hello, my name is Emily Ogawa. I am a senior undergraduate marine bio student, and I did my capstone on invasion of the bubble, spatial abundance of native algae Dictyosphaeria and introduced algae Kappaphycus in Kaneohe Bay. So, for some background on Dictyosphaeria, it is commonly known as the green bubble algae, and it is native to the Hawaiian Islands, as seen on the picture at the right. Older colonies tend to form dense aggregated mats, and younger colonies tend to form small clusters. Kappaphycus is a red algae and it was introduced to Kaneohe Bay in 1974 for the carrageenan industry, and it was used as thickening agents in food and other cosmetic products. And it was done by the Hawaii Institute of Marine Biology for experimental cultivation and research. And algae really likes to grow best when there are optimal light, nutrient, and substrate availability conditions. So the objective of my study was to assess the presence and spatial abundance of invasive algae species overgrowing coral reefs in Kaneohe Bay as the effects of climate change become more visible. My hypothesis was that invasive algae abundance will be higher where environmental conditions are detrimental for corals, and I wanted to look at three factors that could affect algal growth: substrate type, depth, and reef location. So this is a map of my sampling site. Here we have a boundary separating inshore versus offshore reefs, and inshore reefs are also in red, offshore reefs are in blue. And there were 8 total reefs used in this study. And to collect data, we went out on the research vessel Kaholo, and we got in the water and snorkeled to collect our data. And this is a picture of some of my classmates, including my research partner, Christina, and our other partner, Ivy. And this is an example of what it looked like when we were in the water collecting data. For each of the 8 reefs, we did 4 transects on each reef. Each transect was 6 meters long, and we took 12 photo quadrats every half a meter using a half-meter quadrat grid. And at the end of this survey, we had 384 quadrats, and we measured the start and ending depth of each transect and then averaged them and then applied the NOAA tide predictions to correct the tidal data. And this is an example of what one of the photo quadrats ended up looking like. And then we used CoralNet analysis software from UC San Diego to create 25 randomly generated points on our grid. And then we trained the program to recognize things like live coral species, dead coral, and my 2 algae species, Kappaphycus and Dictyosphaeria, both of which are present in this quadrat. And using all of those points, I was able to create percent covers and then apply those for each reef. So this is a figure of average percent cover of live and dead coral at the surveyed reefs. This is the average percent cover of Dictyosphaeria and Kappaphycus by reef number. And then here we can see more of a general pattern, including the 2 substrate types, live coral and dead coral cover versus both of the algal species. As you can see, Dictyosphaeria was present on all 8 reefs, but Kappaphycus was only present on 4. And it looks like there is higher dead coral cover on inshore reefs 1, 2, 38, and 44 versus offshore reefs. And average transect depth was taken to form this depth plot for each reef. And then that data was then converted into inshore and offshore to look at the pattern between reef location and depth. And then this is average percent cover of invasive algae by reef location. As you can see, on inshore reefs for Dictyosphaeria, there is a higher percent cover than in offshore, but note the scale is only up to 2 to 3%. And then for Kappaphycus, there was more presence of Kappaphycus in offshore reefs, but the scale is also greater, up to 15%. So none of these tests that I ran were actually statistically significant, but looking at the trends of the data, if there were more replicates, I think a pattern would have ended up showing. For substrate type, what I did find was that algae was found on reefs with both live and dead coral. Kaneohe Bay is a very heterogeneous environment, and so one reef is not going to be solely alive or dead. There was higher algal abundance with higher percents of dead coral. Depth ended up being more of a controlled variable than I originally had thought it would be, because we always sampled between 0 and 3 meters. It ended up just being a control versus a factor. And reef location, offshore reefs sampled were deeper on average in compared to inshore, and higher percent cover of Dictyosphaeria was seen inshore, but higher percent cover of Kappaphycus was seen offshore. And in conclusion, this is important because algal presence can be an indicator of reef health. Coral reefs are really important ecological ecosystems and ecological engineers, and other studies have even gone far enough to call Kaneohe Bay an exemplary ecosystem for its resilience against both natural and anthropogenic effects. So things like climate change, rising sea temperatures, and ocean acidification are affecting our reef systems, and Kaneohe Bay is such a well-studied system to build off of other historical studies. And it also allows us to get in there as the next generation and provide implications for future studies. So another way that my study could have been better is using a wider depth profile, using more reefs throughout more areas of the bay, and also including more monitoring studies for invasive species if they are introduced to a system like Kaneohe Bay. Thank you. These are my references.